Tēnā koutou katoa, no mai, haere mai, ki te hui, Arati, Aotearoa, New Zealand Food Safety Science and Research Centre. Ko Vivian Hunt aho, ka nui, te mihi nui ki koutou katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science Research Centre, I'd like to welcome you all here today to our monthly Spotlight Food Safety Science Seminar, and welcome especially to our overseas, some of our international science panel, Leon and Arno, calling in today from 10 and 11 p.m. So thank you for joining us today. Today we're really excited to have another two of our students who will share some of their research findings, which follows on from our last Spotlight seminar. So our first speaker is Erin Young, who will talk about her research on smart packaging and whether it can improve the safety of New Zealand food, as well as enhancing shelf life and at the same time meeting the changing consumer demand for authenticity. Erin is a PhD student at the Department of Food Science at the University of Otago. She completed a BTEC in 1997, then a postgraduate diploma in packaging at Massey University. After she started her career in seafood protein chemistry research, she moved into the FMCG industry, and she's now had over 10 years experience in the food packaging with some of New Zealand's iconic brands. Her research will augment her technical packaging experience and graphic design interests. She's a member of the University of Otago's Food Waste Innovation Research Team, which measures food waste and looks at innovative reductions technologies to help companies and consumers change their behaviour towards packaging. So I'd just like to hand over now to Erin, and then after that we'll have Rose Collis, and I'll introduce her after Erin has spoken. So if you have any questions, just put them onto the chat for those of you who haven't attended before, and we'll ask them at the end of Erin's. She'll talk about 15 minutes, 20 minutes. Over to you, Erin.
Thank you. Thank you for everyone who's linking into the seminar, and thank you for that great introduction. My project is looking at Chinese consumer perceptions for using smart packaging on New Zealand food exports. I'm partway through my project, so far having taken a qualitative approach with looking at both New Zealand export industry participants and Chinese consumers. So this presentation is a top-line view of the work to date. When it comes to food safety, packaging is a tool that assists with safe separation through both time and space of food production and food consumption. Food supply chains have evolved over time to meet changing consumer demands. Consumers are currently demanding high levels of product information to support authenticity and promote benefits, and they're also demanding fresher, less processed products with fewer additives and processing. Packaging technologies can step up to ensure that the required functionality continues to be maintained while other attributes like the processing levels are reduced. Smart packaging, which incorporates both active and intelligent packaging, can meet some of those needs. But implementation of innovative technologies such as smart packaging can impact other innovations occurring concurrently. For food packaging, one such competing innovation is the drive towards sustainable packaging. This is particularly driven by global pledges to move plastic packaging to a circular lifestyle by 2025. There are four basic functions of packaging: containment, keep the product in; protection, keep unwanted components out; convenience, making the product easy to use as possible; and communication, which is telling the consumer about the product. Active packaging technologies enhance the protective function. They provide functionality beyond being a passive barrier of conventional packaging. They can absorb or emit constituents that can enhance product quality and safety, reducing the need for other processing technologies. Examples of active packaging include oxygen scavengers, moisture scavengers, and antimicrobial materials. Intelligent packaging enhances the communication function, providing additional information about the history and condition of the product through the use of sensors and indicators. For this work, I've also included cloud-based data management that via a QR code on the packaging provides traceability due to its capability of providing historical information to consumers. Examples of intelligent packaging include time temperature indicators, freshness indicators, and traceability technologies. So why could smart packaging be a good fit for New Zealand food exports? There are a couple of pretty important reasons. The first is that New Zealand is a significant distance to our key markets in Asia, North America, and Europe. And the second is that our common exports are often perishable products like dairy, meat, seafood, and horticulture products. The reliability of packaging and supply chain management around temperature and humidity are critical for export success. The research questions for the exploratory stage of my research were to understand what the influences are on the New Zealand food export industry for adopting smart packaging technologies, and also how do smart packaging technologies make a difference to Chinese consumer perceptions of imported foods like New Zealand exports. To gain insights on New Zealand food export industry, semi-structured interviews were felt to be a suitable method. A predetermined yet flexible set of questions provided a scaffold for conversation with participants without being too prescriptive, so the conversation was kept on target but could explore the unique priorities of participants and their organisations. Interviews were audio recorded and transcribed for further analysis. There were 15 participants that were either technical or marketing or sales based, but all were involved in packaging selection processes and were employed by food industry organisations, packaging companies, or governmental bodies. It should be noted that the participants' opinions were their own and not necessarily reflective of the official position of their respective organisations. The selected data collecting method for the to qualitatively explore Chinese consumer perceptions was focus groups. 3 groups were run in Beijing prior to COVID, utilising a China-based market research company so they could be run in Mandarin. The focus groups were audio recorded, transcribed, and then translated into English for further analysis. There were 8 participants in each of the groups for a total of 24 participants. The moderator presented 10 hypothetical smart packaging enhancements to current New Zealand export products to prompt conversation about their likes, their concerns, and their willingness to pay for the products with the smart packaging enhancements. One such example was a ripeness indicator sticker on avocado. So they were asked to choose between a stickerless one and the one with the sticker. The same data analysis method was used for both the focus group and the interviews. The transcripts were loaded into a software package called Invivo, where portions of the text can be selected and allocated to themes, which can then be reviewed together to gain an understanding of the data. Reflexive thematic analysis was the methodological approach used to generate the themes. Participants in both studies saw food packaging as a tool to achieve functional benefits of quality and safety, as well as epistemic or functional informational benefits to food products. Packaging was also seen as a necessary expense, an environmental burden, and a communication conduit between companies and consumers. Initially, the themes generated were based on literature, with additional relevant themes identified during coding. So on to the results. For the industry interviews, 42 codes were generated during the analysis of the interview transcripts and were organised into a conceptual framework. At the top, you can see macro factors, sector factors, and business factors. These all moderate the relative importance of the benefits and barriers identified by industry. The benefits and barriers, fairly self-explanatory. What did industry feel the benefits were of utilising smart packaging on their products, and what were the barriers to implementation? The relative importance of each led to varying levels of acceptance and use. Overlaying significant parts of the framework were sustainable packaging goals. Moderating factors were on 3 levels. Macro factors affected the whole industry, things like New Zealand's small size and our distance to our export markets. These factors affected all New Zealand exporters. Differences in product type, storage methodologies, and target markets were all sector factors. For example, lamb has different needs from kiwi fruit or dairy products. Markets also have different needs. Several participants discussed the challenge of exporting to China relative to, say, the USA. The size and experience of the organisation can influence uptake of smart packaging as well, and would come under the category of business factors. Participants working for organisations that have been burnt in the past found a higher value in the potential benefits from smart packaging, and bigger organisations had more resources to focus on it. An organisation's leadership can also influence uptake. You can see from the quote on the slide, a board interested in innovation will place a high interest in the benefits of smart packaging. In building the conceptual framework to understand the drivers and barriers to the use of smart packaging, the moderating variables influence the magnitude of impact of the benefits and barriers. Another impact on the uptake of packaging innovation like smart packaging is what else is going on in the industry. At present, another innovation focus is moving food packaging into materials that are compatible with circular life cycle principles. When participants were asked about other packaging related priorities they were working on, sustainability work invariably came up and generated quite animated conversation. It intersects with smart packaging in 3 ways. There were synergies. By improving packaging's protective function, food waste can be reduced. Improving the environmental footprint of food production. But there were also conflicts. Smart packaging can reduce the recyclability of packaging materials. However, the main intersection between sustainability and smart packaging within the New Zealand food export industry is resourcing. Packaging development resource is very much focused on achieving sustainability goals. So unless the need for smart packaging is significant in comparison, it receives a much smaller share of the technical resource pie. Intelligent packaging provides information to the supply chain, or in other words, epistemic benefits. A quote from one of the participants I think was quite enlightening on this topic. They said that, for years people have said packaging is the silent salesman. Well, it's about to get a voice. The participant went on to say that the industry was experimenting, trying new things on a variety of fronts around what information industry and consumers want from intelligent packaging. There were 3 main areas of epistemic value benefits identified in an overseas study that aligned with our participants' insights. They were improved inventory management, traceability for supply chain benefits, product integrity, so that's providing information on quality and safety, counterfeiting prevention, and traceability for the consumer benefit, and then finally improving user experience. Active packaging is usually employed to improve the protective function of packaging, and while the additional protection can assist in inventory management, the primary benefit is to minimise the speed of deteriorative reactions that adversely affect food quality and safety. One example discussed included oxygen scavengers for a high-fat product where oxidation was a significant concern. Having identified the benefits industry participants felt that smart packaging could provide to the organisations, it begs the question of why the commercial uptake isn't higher. What's stopping them? Turns out there's a number of barriers from a larger variety of sources than I had expected. The first one is fairly obvious: cost. The upfront cost of the technology, as well as the perceived added value versus the cost. The example quote was fairly typical, where project work is in progress and then the numbers are run by marketing and the project stops. Legal concerns fell into 2 areas. The first was data management and privacy concerns. Both consumer data, with what they may be providing when scanning codes or linking into data, and also, particularly in the case of traceability technologies, how far do you go in identifying individual farms or orchards as the product source? The other is around regulatory and compliance challenges. An example of that is the Swiss ordinance, which is a positive inclusion list for packaging materials. To be able to use something in food packaging, it must be on the list, which adds to the challenge of implementing novel packaging technologies. Organisational issues included a lack of familiarity with technologies, alternative solutions or a perceived lack of need, or low priority, particularly when compared with sustainability projects, as I mentioned, the lack of available business partners that specialise in needed technologies, and then finally, insufficient technical resource in general to implement projects. Technology challenges also came up. Examples included the commercialisation issues where the implementation of a smart packaging solution slowed a production line down, and also where the technology simply wasn't up to the mark, such as camera technology on the average consumer smartphone to scan intelligent packaging solutions on pack. The final barrier to implementation of smart packaging projects was risk. Risk of the product not functioning as advertised, or that it could cause physical harm to the consumer, like components in sachets that are not fit for human consumption. The final risk was whether consumers would accept the technologies implemented. Despite these challenges, all participants in the study were accepting, to at least some degree, of smart packaging. Utilisation varied as well. Some organisations already using active and intelligent technologies, others interested in their use and pursuing opportunities, while others were aware and theoretically interested, but had other priorities, so were not pursuing any projects. It was suggested in the overseas study I mentioned earlier that the food industry is in an early stage of implementation of smart packaging, and these results seem to validate that finding. As I mentioned, one of the supply chain concerns was consumer acceptance of smart packaging, so it seems a good time to move on to the focus group results. We were able to yield a few numbers. Obviously, with only coming from 24 focus group participants, these are not statistically significant. These results suggest that there is an overall acceptance of smart packaging technologies being applied to New Zealand food exports. And if you look at the pie charts, most participants selected the smart packaging technology over current packaging when given the choice. Willingness to pay also suggested a general acceptance, with the suggested premiums being nearly 11 and nearly 17% for active and intelligent packaging technologies, respectively. Of the technologies that were presented in the focus groups, 5 active technologies and 5 intelligent technologies, the top 3 preferred options were all intelligent technologies, providing information on product security, product freshness, and product authenticity. A systematic review was completed with a conceptual framework developed that could be reviewed against the themes generated by analysing the focus group results. On the far left, you can see the moderating variables and control variables. Control variables were the socio-demographic variables such as age, gender, income, household composition, and so on. Moderating variables are the consumer's broader attitudes to technology, to food, and the environment, as well as trust and familiarity with the technology in question. Both control and moderating variables influence the importance of both consumer perceived benefits and barriers for acceptance, which in turn impact consumer attitudes towards the technology. That forms the attitude half of the model. The model moves into a behaviour intention model where the theory of planned behaviour has been used. Attitude, subjective norm, and perceived control lead to an intent to purchase. In analysing the focus group work, the priority to date has been on the attitude half of the model. With looking at the themes generated, the benefits perceived by consumers were varied, so they needed organising. The theory of consumer values proved to be a good fit, where benefits are separated out into a number of categories and described as either utilitarian or hedonistic benefits. With functions of packaging being primarily utilitarian, it should be no surprise most of the benefits were there. As with the industry perceived benefit, the utilitarian benefits were epistemic value from intelligent packaging and functional value from active packaging. The functional value was an interesting one. On one hand, consumers liked the idea of active packaging to improve the quality of the product, to make it fresher. But when the flip side of quality improvement, as in shelf life extension, was discussed, Chinese consumers were not so keen, citing short shelf life as a measure of quality. Hedonistic benefits for social value, which within the conceptual framework is moved into subjective norms in the behaviour intention part of the model. Emotional value came up and improved feelings of safety, with consumers believing the product and packaging were more reliable. And conditional value is where a product is only purchased if it's needed, such as infant formula is only purchased when you have an infant. In the case of smart packaging, consumers with higher needs for food safety would find packaging technologies that improved food safety important. There were a variety of concerns identified by consumers in the focus groups. The innovation resistance theory was a good fit to organise those concerns. They fell into 2 main areas: functional barriers and psychological barriers. Functional barriers included usage. Technologies that reduced convenience were not popular. The economic barrier was another, where technologies increased the price of the product and the concerns about risk also needed to be addressed. Risk could be broken down into different categories: concerns about the technology itself not being safe, concerns about whether the technology will perform as advertised, and active and intelligent packaging reduces the economic risk of food products by improving food safety or providing information about the product, allowing consumers to have a better chance of consuming the product before it goes off. Social risk and image barrier for the use of active and intelligent packaging are similar and are concerned with the perception of how the use of products with the technologies reflect in the opinions of others' opinions of consumers. Within the conceptual framework, social risk was moved into the subjective norm and the behaviour intention model as well. The final psychological barrier is that of tradition or habit. Where technology demands a change in behaviour or a change in a view, there is resistance. So going back to the research questions from the start, it was established that New Zealand's size, location, types of products businesses export, and some of the business attributes all appear to influence the uptake of smart packaging. Benefits perceived by industry include functional benefits, improving the quality and food safety of New Zealand exports, as well as providing epistemic benefits to consumers around traceability, quality, and security. Barriers were cost, technical resource capacity, and the availability of required technologies. Socio-demographic variables, broader attitudes to technology and food, as well as familiarity and trust, all appear to influence the importance Chinese consumers place on the perceived benefits and concerns they have around industry's use of smart packaging on imported foods. Benefits aligned well with the industry benefits in that consumers could see functional benefits for food safety, but consequently also some did not believe extension of shelf life was a good thing. So communication about active packaging needs to focus on quality rather than shelf life extension. The concerns consumers had were around functional and physical risks of the technology, as well as costs. Overall, the results were positive. Organisations and consumers appear to be interested in benefits, but some of the barriers and concerns were significant. And there appears to be a high level of interest in intelligent packaging. The next step for this work is to quantify the relative importance of some of the perceived values and concerns cited by Chinese consumers. So thank you for coming today. I'd also like to thank MB for the project funding, the New Zealand Food Safety Science Research Centre for my scholarship funding, my participants in both the studies for their time and expertise, the University of Otago, and of course my supervisors, Associate Professor Miranda Morosa, Professor Phil Bremer, and Professor Indra Awe. Thank you.
Great, thank you very much. Thank you very much, everyone. That was fantastic. We have got one question so far. So Leon says, thank you very much for a nice presentation. In the conclusions, you did not come back on the sustainability, waste, recyclability issue. This was not a big deal for consumers in the end? That was one of my questions too.
The Chinese consumers, we did ask them about the environment and sustainability of packaging. I haven't looked too deeply into that as yet. Chinese consumers tend to be more focused on food safety. New Zealanders assume that their food is safe, and so we start to worry about things like environmental sustainability. But Chinese consumers are still very much focused on, is my food safe? So they didn't have strong opinions on that. Yeah.
Maybe it's more of a driver for industry behaviour, is comment. Yeah. I had the same question. So the next phase of your work is to try and quantify some of the responses, and I mean, cost is obviously a big factor. I didn't see costs or price in your model.
It's in the consumer model in terms of the barriers. Okay. And willingness to pay results tend to overinflate the interest. So those sort of figures of 11 and 17% premiums, I think when you actually ask consumers to put their money where their mouth is, so to speak, I suspect they won't be as...
Well, purchase intent's always very difficult to measure, isn't it? I know. Yeah. Is there any other questions from people before we get to our next... you could send them afterwards. We'll have some more time afterwards, just quickly. Any further questions from anyone? It doesn't look like there are at this point. So yes, I think I thought it was a great talk. Thank you, and we'll get some more questions later. So just hold on, and if you could just switch over now, and Rose will share her screen. So thank you, everyone.
Thanks.
Thanks. And we'll talk soon. Rose, will you share yours? So while Rose is getting ready to share her slide deck, I'll just talk to you a little bit about Rose. So she's going to talk to us about her work on antimicrobial resistance in dairy farming. So Rose Collis is a third-year PhD student with the food assurance team at Ag Research and the EpiLab at the School of Veterinary Science at Massey University. Rose was born and raised on the dairy farm near Ikirahuna. She completed a BSc and Masters in Science at Massey University, where she majored in microbiology. During her undergrad studies, she held 2 summer internships at Ag Research in the food assurance team. So her PhD research focuses on the prevalence and distribution of antimicrobial resistance in 2 New Zealand dairy farms, and uses both culture-based methods and shotgun metagenomic sequencing techniques. So I'll hand over to you, Rose, and you can get started now. Thanks very much.
Well, thank you for that great introduction and for the opportunity to present to you all today. So I'll move straight into my results after that really good introduction and some background. So my research is focusing on antimicrobial resistance. As you're all aware, this is a global public and animal health concern. The misuse and overuse of antimicrobials in both human and animal health have been suggested as the main drivers of resistance. However, the development of antimicrobial resistance is complex, and other factors such as heavy metal, biocide, or disinfectant use may also select for antimicrobial resistance. So this figure here is from a report published in 2014 that aimed to estimate the burden of antimicrobial resistance. And it was estimated that at current rates, by 2050, 10 million human deaths per year will be attributable to antimicrobial resistant infections. So this is a global public health concern. The transmission and development or spread of antimicrobial resistant bacteria is complex. So this figure here is showing potential transmission pathways. So these orange arrows, which is the misuse or overuse of antimicrobials, has been suggested as one of the main drivers of resistance, and the green arrows indicate potential transmission pathways. So as you can see, it takes a One Health approach when you're looking at antimicrobial resistance, because there is interactions between humans, animals, and the environments. So this is looking at from a dairy farm perspective. So we have the cattle, which may have resistant organisms that can be excreted in the feces and go through the environment back to either the animal or humans. In terms of the dairy farm, there's also food, animal-derived products, which would be milk. So in New Zealand, the milk is pasteurised, so that's really low risk, but it shows all of the factors when we're looking at the farm environment. In terms of antimicrobial use in animal production in New Zealand is a relatively low user. So a report that was looking at antimicrobial sales data from 30 countries from 2012, so this data is a little bit older now, showed that New Zealand was the third lowest user of antimicrobials in animal production, and actually used 12.9 times less than in human medicine. Although this is low use, antimicrobial use and the development of antimicrobial resistance in food producing animals is a growing consumer and regulator concern globally. There are a number of resistant organisms which are of concern. My PhD is partly focusing on extended spectrum and beta-lactamase and AMP-C producing Enterobacteriaceae. So what are these? ESBL and AMP-C are beta-lactamase enzymes, so they can hydrolyze beta-lactam antibiotics and they can confer resistance. So the ESBLs are resistant to a number of antimicrobials shown here. So that's the penicillins, monobactams, and first and third generation cephalosporins. The AMP-C beta-lactamase enzymes have a wider spectrum of activity, and they're also resistant to the second generation cephalosporins and the kefamycins. So this next column here is talking about transmissibility. So what does this mean? Often ESBL beta-lactamase enzymes are encoded on plasmids, and this is of concern because plasmids can be shared between bacteria and even within bacterial species. The AMP-C beta-lactamase enzymes, they can either be encoded on the chromosome or they can be plasmid mediated, and this is also species dependent. The World Health Organization has a pathogen priority list, and ESBL producing E. coli Enterobacteriaceae have been listed as critical on this list. One of these reasons for this listing is that ESBLs are often associated with other genes that code for other types of resistance, so which can result in a multi-drug resistant phenotype. In terms of the agricultural environment, the global detection of AMP-C and ESBL producing Enterobacteriaceae has been found in a number of agricultural environments, including dairy, poultry, and swine. These figures for some prevalence studies are from overseas, and what I wanted to show you here is that there is often a wide range of prevalence between studies, and that's because it's really hard to compare between different studies because it can depend on the study design, some of the methods they use to actually look at these AMP-C and ESBLs, what type of farming systems, so whether it was overseas and other factors such as the antimicrobial use or whether it's more intensive farming. So that leads into my research project. So I wanted to look at the prevalence of AMP-C and ESBL producing Enterobacteriaceae in New Zealand dairy farm environments and look at the prevalence of antimicrobial resistance genes in these two New Zealand dairy farms. And my hypothesis is that because of the low antimicrobial use, we will have lower levels of resistance. So the two research farms used in my study were the two Massey University dairy research farms. One is a smaller farm, which is less intensive farming, and the second farm is actually a larger farm that's used to study more intensive farming practices, such as the use of an indoor barn. So I collected samples each month over a 15-month period, and that's so that I could capture all the changes over a dairy farm. So each month I collected samples such as the raw bulk tank milk, effluent, soil, supplementary feed, fecal samples, any bird poo that I found on the farm, and importantly I also talked to the farmers about management practices, antimicrobial use, and anything else that might have been happening on the farm that could influence antimicrobial resistance. For my sample screening, initially I've just focused on the soil, faeces, bulk tank milk and effluent. And this next slide here shows the sample screening actually trying to isolate these AMP-C and ESBL producing Enterobacteriaceae. So I took all of my samples back to the lab. They were enriched overnight, and then this enrichment was plated onto 4 different types of agar. So the first is McConkey agar, that's selective for enteric bacteria, and that was also used as a control to ensure that there was growth in the samples. The next 3 agar were selective for the AMP-C and ESBLs. So the first 2 are McConkey agar with 2 different 3rd generation cephalosporins added, so these are antimicrobials. And the last agar is a commercial chrome agar specific for ESBLs. So from my screening, I found 32 E. coli from faeces and 20 from effluent from these selective agar, and I didn't find any on these agar from the soil or the bulk tank milk. I then went on to process my isolates, so looking at the species identification using multi-TOF and mass spectrometry. So I was mainly interested in either E. coli or Klebsiella. I then wanted to do antimicrobial susceptibility testing to look at the resistance profiles of these isolates. So what this means is here's a bacterial plate, you can see. So this here is the growth of the bacteria all along the plate. We can add these antimicrobial discs seen here. And then based on the zone size, and there's guidelines which depend on which antimicrobial used and which bacterial species it is, you can see whether the isolate is susceptible or resistant. So for example, this antimicrobial here, you can see there's a really small zone size, so the bacteria can grow really close to the disc, and this isolate is resistant to this antimicrobial. Whereas this one here, you can see there's a large zone where the bacteria hasn't been able to grow, and this bacteria is susceptible. So for this, I used 6 different discs. The first 3 were beta-lactams, so that was looking specifically for the AMP-C and ESBLs, and the others were 3 discs of clinical significance. So these are the results of the antimicrobial susceptibility testing, with the 6 antimicrobials along the bottom and resistance shown in red and susceptible in green. As you would expect, most of the isolates were resistant to these first 3 antimicrobials. So that's not surprising because all of these isolates came from the were selected for on the antimicrobial agar. And there was reasonable levels of resistance to both tetracycline and streptomycin. However, all of the isolates were susceptible to ciprofloxacin. I then went on to take any isolates that were resistant to these 2 antimicrobials to confirm whether they were ESBLs, and anything that was resistant to the cefoxitin was confirmed whether it was an AMP-C positive. So this confirmation testing is using phenotypic methods, so they're commercially bought antimicrobial discs. For this one here, for the AMP-C confirmation test, we have 3 different discs with a combination of an antimicrobial, an inducer for AMP-C, an ESBL inhibitor, and then an AMP-C inhibitor. Similarly, the confirmation test for the ESBLs, there's 4 different discs, so 2 different antimicrobials, and then the antimicrobial with clavulanic acid, which actually is an inhibitor of ESBL. So for example, these 2 discs are the same. This is just the antimicrobial. You can see a small zone here. This one is the antimicrobial with the inhibitor, and you can see a large zone I then wanted to look at some of my isolates in more detail. So to narrow it down, I did some molecular work. The first was E. coli phylotyping, so looking at the E. coli population diversity. E. coli has 8 phylogroups, and I used the Clermont typing method. So this initially looks at 4 different target genes. Based on the products of this PCR, you can use a decision-making tree to work out which phylogroup is present. So along this graph on the bottom, you can see the 6 different phylogroups found in my study, and the number of the isolates were from phylogroup B1 and C. I then wanted to do some characterization of the AMP-C genes. If you remember from the start of my talk, the AMP-C gene can either be encoded on plasmids or encoded in the chromosome. So initially I started by looking at the plasmid-mediated AMP-C genes. So this is using a PCR. There's 5 main groups based on sequence similarity, and the BLA CMY2 gene is the most prevalent globally. If the isolates didn't carry a plasmid-mediated AMP-C gene, I then wanted to look at the promoter region of the AMP-C gene in the chromosome. So E. coli normally express AMP-C at really low levels. However, you can have mutations in the promoter region of this gene, resulting in a stronger promoter and therefore hyperproduction of the AMP-C beta-lactamase and resistance. So this is a little summary of my isolates. So AMP-C down the bottom is chromosome-mediated resistance. AMP-C with the P is plasmid-mediated, ESBL, and then unknown is an unknown resistance mechanism just based on the phenotypic testing. So you can see a larger number of plasmid-mediated AMP-C on the secondary farm, which is the more intensive farm. So this next slide here shows the distribution of all of the 52 isolates, and I've selected 12 of these shown in yellow to undergo whole genome sequencing. So these have been sent away so that I can better understand the genomic epidemiology of the AMP-C and ESBL producing E. coli on the 2 farms. Now looking at the second aim of my research, which is looking at the antimicrobial resistance genes within the farm environment. I've used the same 4 main sample types: soil, feces, bulk tank milk and effluent, collected from the 2 dairy farms over 15 months. And then these samples underwent shotgun metagenomic sequencing. So what this means is you take a sample, for example, soil, you extract the total DNA, and then this can be sequenced, so you have sequencing reads matching the total DNA from the sample. Because our hypothesis is that the antimicrobial resistance genes will be at a really low proportion in the samples, we wanted to sequence at a high depth to capture this information. So I now have more than 11 billion DNA sequence reads across my 4 sample types. So just to put that in perspective, for every single sequencing read, if it was a piece of paper and it was stacked on top of each other, that would be 880 kilometres high. So that's the same distance from Wellington to Kerikeri. So I have a lot of data that I'm currently going through. So I've just got a couple of preliminary data slides. So this is based on a subset of the reads, so 2 million reads per library, and we have on average around 40 million reads. So the reason we're going through a subset is just testing all of our processes to make sure they work before doing it on the full data set. So one of the steps I've been going through at the moment is normalization. And we want to do that because there can be bias when you're comparing abundances of genes between samples when they have different numbers of microbial DNA. For example, comparing a sample such as milk, which has really low microbial DNA, versus something such as feces, which has high levels. So these first 2 graphs show 2 different methods. This first one is looking at genome equivalence. So that works out the average genome size in a sample, and that's relative to the base pair sequence, which will give you your genome equivalent. The second method is using the 16S rRNA genes. And as you can see, this graph down here is the comparison of both methods, that they worked relatively well. There was clustering based on the sample types close together, with a lot more variation in the effluent. This next slide here is again just on a subset of reads, looking at the relative abundance of taxa. So that taxa along here. So this could be, for example, a specific bacterial species. And then the heat map is based on the fraction of total reads, with red being lower level and blue being higher. And again, what I want to show you with this figure is that the clustering is based on the sample types, as you'd expect, with some variation between samples and also a lot more diversity found in the effluent samples. So that leads me on to the future work of my PhD. So as I mentioned, sent 12 E. coli away for whole genome sequencing to get a better understanding of the genomic epidemiology of these isolates. I'm still analysing all the management practices and antimicrobial use data between the two farms, and the analysis of the shotgun metagenomic sequencing is still ongoing. So this is looking at things such as antimicrobial resistance genes and the bacterial community composition. So in summary, there was a low number of AMP-C and ESBL producing E. coli isolated on the two New Zealand dairy farms. 33 of the 52 isolates were multi-drug resistant, so that means they were resistant to more than 3 classes of antimicrobials. 28 of the 46 AMP-C producing E. coli were plasmid mediated, and the shotgun metagenomic sequencing, the analysis is still ongoing. So I'd just like to finish with some acknowledgements. So my supervisors, Adrian Cookson, Patrick Biggs, Anne Minritter and Sarah Burgess, and a number of other people who have given advice during my study, the farm managers on Dairy 1 and Dairy 4, and especially to my funders, so the New Zealand Food Safety Science and Research Centre for my PhD stipend, the Ag Research Food Integrity SSFI program, the School of Veterinary Science Postgraduate Research Fund, and the Eric Ajala Trust Postgraduate Scholarship. So thank you all for listening.
Great, thank you very much, Rose. I've got a couple of questions here. So one question is, do you know, are these antibiotic resistant genes encoded in the genomes a part of some type of genetic element, or are they non-mobile genetic elements?
Yeah, so the slide that I showed, oh, I didn't show that one first. So we're doing a couple of things. So the first one is we're looking at the sequence reads. So that's just mapping to a database of resistance genes. So that's just looking at whether they're present or not. The second thing we're doing is we're assembling the reads into larger contexts of DNA. Then we're looking at resistance genes within these, and then we can do taxonomic classification to find out where that resistance gene is, so we can look at it in more detail to see whether it's, or where it's encoded and which bacterial species it's in.
Okay, so at this point you don't have an answer on
whether no, at the moment we've just got all of the data and getting the pipeline set up, and then once it's run, I'll hopefully be able to tell that. So that's the plan, but not yet.
Excellent. Leon has also said, thank you, Rose, for interesting presentation on antimicrobial resistance transmission. Here's a question about communication. What he understands correctly, all resistances that you study were caused by antibiotics used for food animals, human health. So why not call it antibiotic resistance, ABR? And then obviously there are some other antimicrobial chemical aids that are used in food production and processes, but these have a different mechanism of efficacy and possible resistance as compared to antibiotics. Is more specific terminology not better to communicate about where and what the ABR genes, risks and mitigation options are?
Yeah, so that's a good point. So for the culture work, I have focused specifically just on the AMP-C and ESBL types of resistance. For the shotgun metagenomics, we're looking at everything. So some of the databases I'm using have antimicrobial resistance genes, but also biocide resistance, heavy metal resistance, and all of those factors involved. But that's a good point to think about when I'm talking about different aspects of my project.
That's great.
Thank you.
And Roger Cook has asked, he says, great talk, Rose, huge thanks. Wants to know how representative of New Zealand dairy farms are the two Massey Research dairy farms?
Yeah, so that's a really good question. So the two Massey farms were chosen for a number of reasons. We looked at whether I should sample a large number of farms across New Zealand, maybe just once, or whether I should focus on two farms in a bit more detail. So that's why I wanted to do the two farms so that I could capture all the changes over the time, such as changes in antibiotic use, calving, other factors. They are good farms in terms of they often have the vets for the antimicrobial prescribing, but the main comparison was between the two different management factors, so one being a more intensive farm and one being a lower intensive farm. but it's, I think, in the way I interpret my results, I'm not expecting my results to be representative of all dairy farms in New Zealand, but just focusing on these ones specifically.
That sort of links a question I had. You said that you looked at studies on prevalence, mainly global studies, which influence results between 13 and 94%. Is there many prevalence studies in New Zealand?
There's not too many prevalence studies in New Zealand. There is a few from other members in our group looking at a smaller number of farms just once or twice, but it's a really important factor when you look at the prevalence. So I did a review of ESBLs and New Zealand dairy farms, and there's some tables in there actually looking at the number of farms screened, the sample types, their methods, because when you're looking at the prevalence, you've got to take into account all of that info as well.
Okay. Now Arno has asked a question. He's a specialist in this sort of area. I think there's a lot of different ESBL, etc. genes which are shared by different bacteria. Will you check for E. coli specific, or is the presence of ESBL, AMP-C in any bacterium enough, i.e. only focus on E. coli or other Enterobacteriaceae?
Is for the culture work or the metagenomics? Because for the culture work, I haven't checked which specific SBL genes they have yet, just because I'm using whole genome sequencing. So as soon as I get that data back, I can see that. But for the shotgun metagenomics, we're going to look at everything, and I guess depending on what we find, we might hone in on a bit more specifics.
That's great. Does anyone else have any other questions? Guess you've got a couple of experts in the field on the call, so I certainly learned a lot. Everything's advanced. I mean, didn't know about shotgun metagenomics, so it's an interesting area for me. Yeah. And I'm sure a lot of people have learned a lot too. So any further questions from anyone, or do you have other questions about the first talk from Erin?
Can I just continue on my question briefly?
Yes, Arno, do that.
So if you find ESBL genes or AMP-C or anything in other bacteria, that is not necessarily means that there's a problem on the farm. So you may actually get a false positive, and you may take action that is not warranted. So you may be sort of crying wolf if you do that. So this is why I think it's important to actually know in what background it is, because an ESBL gene on its own may not necessarily be problematic. It's only problematic if it's in bacteria that could get into the food chain or cause infections. So this is why I'm also a bit cautious with over-interpreting metagenomics data.
Yeah, definitely. That's a really good point, and that's why we're hoping to do actually assemble the reads into the larger contigs, and then look at which contigs have the resistance genes, and then do taxonomic ID on that. Because you're exactly right, you need to look at the context of the resistance gene. Just because it's there, it's which species. Is it functional as well? So that's a really good point, and something to keep in mind when communicating the results, because you don't want to overinterpret too much.
Because that will work when it's in a genomic position, but if it's on a plasmid, that won't tell you which bacterium it is. So again, I just want to, because you say if you report it as a positive, they may need to take measures, and those measures may have a significant economic cost, and you only want to do that when there's really a problem.
Yeah. Yes. Yeah, no, that's a good point. And I guess too, we're expecting these to be in a really low proportion. Just the number that I've isolated from the farms is actually a really low. So comparing it to the other studies, whether it's a big problem, I think my hypothesis is that New Zealand dairy farms do have really low levels. So that's something to keep in mind. Yeah.
They'll love that.
Yeah. Any further questions? Arno, do you have any more? Leon, rather?
Could I ask another question to Erin, please? You mentioned then that a marketing company did the focus group work with the participants. Must have been quite difficult for you maybe to instruct the marketing company how to do the focus studies, especially how to explain the technical part of the various technologies that you have been looking at. How did that actually work in practice? It must have been not too detailed, but sufficiently detailed for the consumers to understand what was kind of the functionality, but also what were some of the maybe impacts that are not necessarily the benefits.
Yeah.
Or was there only very technical brief done?
We didn't give the participants too much technical information because we wanted to get their impressions based on the kinds of levels of information they may get, going to the supermarket kind of thing. So so we produced a moderator's guide and everything, including a slide deck in English, and then as part of the contract they translated that into Chinese, and then we sat down and we talked through it with them the day before we did it. We went over to China to do it, and so we sat down with them and just went through it and made sure we were on the same page in terms of some of the translations. And then each of the technologies was presented just as a photo, like the one that I had in my slide, and they were asked to make a selection, and then they were given some information about what it was. So some of the conversation was then guessing and discussing possibly what it could do and why it was there, because they wouldn't have information when they're in the supermarket. So then we explained the technology, and then we asked them again, do you still like it? In most cases, the acceptance went up. There was one example where the acceptance did go down a little bit, and that was around that tradition barrier and perceptions of naturalness. They felt that the active technology on that particular one was reducing the naturalness of the product, so it made it less desirable for them. And then at the end, we asked them to pick their top 3. So yeah, it was really around just generating that conversation.
Okay, well, thanks for explaining that.
So was the focus just on the avocado ripeness sticker?
That was one of the 10 examples. So we had an active technology and an intelligent technology for dairy, for meat, seafood, and horticulture. So we tried to cover a range of different technologies because different ones might have different perceived risks, and a range of product types that we commonly export from New Zealand.
And Midwinter says, do you want to totally pay for an avocado ripeness sticker? I would too. Yeah. Oh, that's great. Well, I think on that note, I think we're running out of time, and appreciate we've got 2 people on late evening. Thanks everyone for calling in. Our next seminar is going to be on the 27th of November, which will be by Dr. Denise Conroy, who's a social scientist at Plant and Food. She's going to be talking about food safety, consumers, and novel technology adoption, and she's going to share the research of her findings from research that she's done both in New Zealand and in China around attitudes towards food safety, and especially from a consumer's perspective. So yeah, that's great. So thanks everyone for calling in today. Just check that I haven't missed any messages. And yeah, so thank you Leon and Anu for taking the time, and great presentation, both Rose and Erin. Thank you.
Thank you.
Thank you. Thanks everyone. Bye.